DNA cez mRNA až po proteíny. Veda je všade okolo nás. Vítajte pri špeciálnej epizóde Vedátorského podcastu, kde sa rozprávam s Emmanuelle Charpentier, nositeľkou Nobelovej ceny, ktorá je známa kvôli svojmu objavu technológie CRISPR-Cas9 alebo ľudovo povedané genetických nožníc. To je vlastne tá technológia, ktorá vám umožňuje editovať gény, ako keby to bol textový dokument. Vystrihnúť nejakú príslušnú časť a nahradiť ju inou časťou, takže napríklad je nejaká porucha génu, funguje nesprávne, vycvakne sa a nahradí sa správnou verziou. Alebo môžete napríklad zobrať baktériu, vložiť do nej gén, ktorý vytvára inzulín a hľa, baktérie budú robiť inzulín. Niečo na tento štýl. Takže CRISPR-Cas9 asi najvýznamnejšia genetická technológia, aká bola objavená, bude formovať aplikácie genetiky v najbližšom storočí. A keď som sa teda dozvedel, že Emmanuelle Charpentier bude na Slovensku a budeme spolu rozprávať v rámci ESET for Schools, čo bola vlastne taká akcia deň po ESET Science Award, ktorej komisii predsedala Emmanuelle, tak keď som sa dozvedel, že obaja budeme rozprávať na tej akcii na ďalší deň, tak som sa jej opýtal organizátorov, či by sme s ňou nemohli nahrať podcast. Oni boli najprv, že jasné, môžeme. A potom ale prišli komplikácie, Emmanuelle mal trochu problémy s hlasom, obmedzila počty mediálnych výstupov, že aby si šetrila hlas. Ešte keď som jej v ten deň stretol a pýtal som sa len, že ako sa má, tak prvé, čo mi povedala, že šetrí hlas, že ho trochu niečo, neviem, obišla nejaká choroba, to už som sa nepýtal. Tak som aj Jozefovi písal, že aká je situácia, že možno sa jej opýtam, že či by s nami niečo predsa len nenahrala, ale ju bol ten hlas. Jozef odpísal, že že kašleme teda na to. A keď sme mali obaja po prednáškach, my sme boli prví dvaja na rade, tak som sa teda opýtal, že či by 5-10 minút len nezodpovedala na pár základných otázok. Sľúbil som jej, že sa nebudem pýtať, čo je to CRISPR-Cas9, lebo predtým hovorila, že ona už dala asi 2000 rozhovorov, kde to vysvetľovala a vedel som, že to je najhoršie, keď už človek len musí na takého autopilota odpovedať. Tak som sa jej namiesto toho spýtal, či je spokojná s tým názvom a tak ďalej. A nakoniec nás museli v podstate až v úvodzovkách vypnúť. Tak sa rozprávala, že cca pol hodinku to má. Možno občas budete počuť zvuky. ESET for Schools prebiehal v Slovenskom národnom divadle. Prišlo tam stovky školákov a my sme boli v backstage. A dozvedel som sa, že keď ste v backstage v divadle, tak vám občas oznamujú, že pozor, teraz končí táto časť programu, asi aby sa nejaký herec nezabudol niekde, neviem, na záchode alebo čo. Tak nám tam občas niečo zahlasia. Tak sorry za takúto trošku zníženú kvalitu, ale Emmanuelle Charpentier mi prišla ako neuveriteľne inšpiratívna osobnosť, veľmi ľudská, veľmi hlbavá, zaujímavo odpovedala na otázky. Často inak, ako som čakal, ťažko sa vedie taký rozhovor, kde vôbec netušíte, akým smerom sa to stočí, ale pre mňa osobne teda veľký zážitok. Takže nech sa páči, Emmanuelle Charpentier.
I promise I will not ask you what is CRISPR-Cas9, but are you happy with the name?
Yeah. CRISPR-Cas9. Am I happy with the name? So CRISPR was a name that was given by my colleagues. Mm-hmm. Specifically, if I recall it well, Morica in Alicante. Actually, I think it sounds like a funny name. But I like it because it's a little bit like crispy, CRISPR. So it sounds like crispy. At the end of the day, you see a lot with regard to food. You see a lot of CRISPR because you have a lot of crispy protein bar, crispy salad, crispy whatever.
So people think this is from GMO.
Yes, maybe, maybe. No, but I think it's nice, CRISPR. And I think it's interesting because for a name, it's interesting that it's something that is a bit special, but yet that can refer to something, interesting.
People know how is the backstage of events like this.
Exactly.
Because we're recording during the ESET for Schools, which is part of the ESET Science Award.
Yeah.
So just people listening to the podcast know. In physics, we are terrible with picking names. We say like flavor and color, and about when we are speaking about quarks, nothing to do with this stuff. So, as you said, the name is finally set. You're happy with it, and I think second half of the previous century was mostly about understanding genes. Like, what the hell is going on? From Schrödinger's What is Life up to this point when we can read human genome, many different animals nearly completely understood. And I think the next century will be mostly about understanding the gene expressions and also manipulating genes. So are you interested in both of those topics, or mostly how to manipulate with genes and you don't care about gene expressions?
No, I'm actually more interested in really understanding the functions of genes, because, even in microorganisms like bacteria, we know only for 1 or 2% of the genes what they are really doing. Mm-hmm. So the rest is really a full mystery. Like for the viruses of bacteria, because this is what I refer to shortly in my talk. These are pieces of DNA integrated in the DNA of the bacteria that are kind of big, actually. And we know only the function of 2% of the genome of the virus. So I think there are still a lot to understand about genes and their expression. After, the tools are important because these are the tools that allow to really understand the mechanisms of life. Having said this, for CRISPR, what is interesting is that it's because I was trying to understand the functions of genes that I came up to this system, which ultimately can be harnessed as a technology. And this is often the case when scientists work on bacteria, and this was always my leitmotif and always what I was, let's say, the way I was justifying the money that I would need for my research was that to understand certain mechanisms, understand the functions of genes and deciphering new mechanisms could allow either to find mechanisms that could be harnessed and developed into gene technologies, or find mechanisms that will lead to a new way to develop anti-infectives, meaning new ways to develop drugs against bacteria, because, as we are very happy to have had a vaccine against SARS-CoV-2, it's another story with bacterial infection. So, we hope, I hope and we hope that we will not have to deal in the future with a pandemic due to a bacteria, because it's more challenging to find treatments.
Mm-hmm. Yeah. So the view that, well, I'll just cut out this part of the gene and sweep it for different elements. Voila, I will have brown eyes. This is overly simplistic because it's so interactive and very complex. The Nobel Prize this year was for microRNA, which is also full in this gene expression. Do you think you're reaching a point when this is just too difficult for humans and we'll just ask AI, like, this is a huge data set of genes, this is a huge data set of different aspects, and go figure out and let us know what should we change? And shouldn't this be very dangerous?
Yes and no, because AI in biology is what we call bioinformatics, and it has been used for years. Mm-hmm. And without the bioinformatic tools, maybe I would not have presented today research on CRISPR, because the research on CRISPR, for sure, I had a very, very short time to explain, but it's a largely evolving system existing in microbes. And we understood this diversity of systems exactly through bioinformatics, actually. And the bioinformatics gave a clue of what could be the function of this system through understanding that there were some genes involved that will code for proteins that would have some motifs that were resembling motifs of proteins which were already described before to interact with DNA and RNA. So AI can really help a lot the biologists. They can help a lot, let's say, anticipating further what can be the function of genes. I think it's like AI. AI can be extremely useful as long as it's machine learning. So it's always about what you feed into the machine, you know. So, but if the machine is fed by solid experimental data and solid data from the past that have been verified, then the machine can learn and can come up with, a lot of solutions. Then after, I'm not worried in this case because I would be worried if people take the AI, let's say, the AI that is more generative and very, as granted and saying, okay, AI told me this, so it is like this. No, but, letting AI help us and for example, for the use of CRISPR as a gene technology, in a way, AI is also important because it helps to design the right RNA to program correctly the technology. So it's very helpful. But again, you need the human beings behind to verify that, what AI told you, it's indeed, true. What I found fascinating with the Nobel Prize, it's also, there is microRNA, which I did not understand actually. Why microRNAs? Because there was a Nobel Prize given for small interfering RNAs 18 years ago. And for me, this Nobel Prize should have been given earlier. It should have been given maybe right after the small interfering RNA discovery, because it's both actually Nobel Prize awards at Nobel Prizes at 18 years difference reward research that was published at the beginning of the 90s. But I think it's good because at least maybe it's not bad because people are reminded again that RNA is important. But I'm very excited by the two other Nobel Prize of AI.
Physics
and chemistry. That is, again, like CRISPR. I'm not a chemist, I'm a microbiologist, but it's a prize for chemistry, and AlphaFold as well. And AlphaFold is the same thing, they worked very hard on the software, and now it allows to predict very nicely. It doesn't mean that the experimental data that use crystallography and cryo-electron microscopy to really show indeed that the structure is as such is not, required for validation. But when one wants to go very fast in research, when one realizes that de facto for certain findings, you cannot do biological experiments at large scale for everything, and then the AI and bioinformatics can be useful to give an idea and help, going through the big data.
Mm-hmm. And AI is maybe the single example of a technology that hasn't been rolled out yet completely, but we are discussing a lot. We at least understand some of the risks and try to incorporate into our lives. Historically, with nuclear energy, we are behind. Like the discussion came decades after people start not being happy about something, which is very unfortunate because, now people are very scared of the technology that is very, very helpful. And I think this is same for gene modifications. It's a very useful technology and people are very scared. So do you think we scientists will be able to, proceed in the future to develop the technologies, or people will be just too scared if we don't communicate clearly and help them understand what's going on?
So what you said, it's exactly right. And it's for AI. AI started in the 70s. It's the same for gene technologies. It started in the 70s. And so they were already concerned at the time. And there were already, conferences organized to discuss as to whether one should put some guidelines in place to, to anticipate the misuse of the gene technologies, like any misuses of technologies. But having said this, there was at the time maybe a threat more for bioterrorism, the idea to modify a microorganism that could indeed, be spread in the planet and provoke a pandemic, etc. But actually, we have not seen this, that is one point. We have not seen this over the past 56 years. For SARS-CoV-2, there were some conspirations theories at the beginning, but everyone has came to the point that no, it was, viruses evolve very fast and this was a natural process. But having said this, I think the gene technologies have been used with care and respecting the regulations so far, including CRISPR over the past 12 years. The fear has been here at the beginning, let's say, has been revived because it was a technology that was new. So it revived the questions that were asked already, 50, 60 years ago. But de facto, when you look after 12 years, the technology has been used very nicely. Only one case was extremely not wanted and quite dramatic, was in China in 2018, a Chinese scientist who came to use the technology to create so-called CRISPR babies. Mm-hmm. With a mutation in a receptor of a T cell that would allow the kids to be HIV resistant, which was heavily criticized because there were publications released that showed that if one introduced this mutation, one could also lead to more susceptibilities to other types of infection.
The complexity and interaction.
Exactly. And this was, so this has been, more than criticized. This has been, by the scientific community, by academies, etc. So, and de facto, the scientist has gone to jail. He came back since and still continues research on CRISPR. So be it. But, having said this, there are always risks. For sure, in 100 years from now, we do not know whether the technology could be used to a certain type of human transformism, but I doubt it because a lot of characteristics are multi-genetics. I mean, it's not caused by one gene, but by multiple genes, and it's not as easy as it sounds like to correctly in a safe manner, without any risks, to really modify genes in the human germlines in a safe manner when it's more than one gene. So it's not as easy as it sounds like. And also I believe that there are other problems on the planet and that at least to use a technology which, is also a possible use for, let's put it that way, counteract certain mutations that are causative of diseases. There are other possibilities with in vitro fertilization and the selection of clones in vitro to go through this procedure in a natural fashion, in a way, by a prenatal selection, rather than doing really gene modification. And I think the scientists are working, following regulations of ethics in the laboratories, which also, allows to minimize the risk of the technology.
So I'll have two final questions. One is regarding the future of humankind, and the second is regarding past of yourself. So a very opposite question. The first question, maybe simple, maybe very hard, is what do you think will change the face of humanity at the end of this century more? AI or gene modifications?
Or AI.
Yeah,
surely, definitely. I mean, gene modification, AI, everyone uses AI. Not everyone is using gene modification and will use genetic modification. So clearly, AI. AI is, let's put it that way. I see AI as a tool in the future, as a tool that we use every day and that impacts us constantly. Gene modification will have tremendously helped the field of life sciences, understanding the mechanisms of life and leading to more treatments in health, more cures, and leading as well to the production of safe crops, which will be, more and more needed in the future. Yeah. So you will tell me So you will tell me maybe people will be confronted daily to it by eating plants that are, produced by CRISPR. But by the way, plants produced by CRISPR does not mean, bad plants. It means that you can recreate using CRISPR actually crops that were produced in the past. So it can also have a positive effect going back to the natural plant that people were using before. But having said this, I think AI has already transformed our lives and will continue to transform our lives.
You touched upon an interesting
and
subject.
it's much more uncontrollable. There is more of a risk of uncontrollability than gene modification when you need the setting of a lab. Not everyone can, AI as well, you need experts in a lab. But what I want to say is that it can really spread. And you see, if we refer to ESET, it's a cybersecurity company. It will require a lot of, development of security around AI, not only IT, but AI.
So hopefully we want to reach a stage when you can just order a do-it-yourself kit with a virus and something you can just, make it more, resistant and so on. And you touch upon an interesting subject that, we can revive old plants, old animals, mammoths, so on. But this is a nice thing also about living organisms, that our DNA is encoding our past. Like the thing that we evolved from apes and we used to live in the ocean, it's somewhere hidden in our DNA, which is, for me as a physicist, absolutely magical. Like the history encoded in a simple molecule, still being present and still like taking a snapshot of the history of life on our planet. So my last question is about your past. What brought you into science? And now you give a lecture to hundreds of students during ESET for schools. And hopefully some of them will be brought to science as well. So what brought you to science, and what would you recommend young people how to pursue career in science? What is the important thing they should have?
So actually, I don't know really what brought me to science, but there are some aspects, obviously, in my life that must have been important. At least a teacher when I was 12 years old, because my mother relates to the fact that I was coming back one day, and I said, when I was 11, 12 years old, and I said one day I would work at the Pasteur Institute, which I ultimately did. But she reminded me this the day where I told her, actually, I found a host lab at the Pasteur Institute. I think most likely also my mother was working in psychiatry. My father was responsible of parks and gardens in the city, so he knew a lot about plants. My mother was more in psychiatry, so it was related to health. And so most likely this has maybe, influenced me in some ways. But I think it was maybe more curiosity. I was interested at some point in all aspects of sciences, could have been also human sciences, sociology, philosophy. I think physics as well, actually, and mathematics. I think I was quite good in mathematics. Maybe I should have chosen mathematics. But the fact is that I think for me, what brought me into science is more the process of having understood at an early age as well. This was referring to a big difference of age between my older sisters and me. When I was entering primary school, she was entering university. And I understood that there were some people who were staying at school all their lives. But not the school for the kids, like your teacher for a school for a kid, but also at universities where it was like, you do research, you're more on the high sphere, and like a kind of, sect, or, And I was finding this very nice to say, okay, you can live your life by asking questions, by, like the intellectual part of the endeavor science that was attracting me, actually. And I think that's why I went to science. It was because I was finding it more interesting than maybe, another job. And also because my parents wanted to study. I mean, they were born at the end of the 20s, beginning of 30s, last century. And I think they were from, a very humble background, and they wanted to study. They could not study. Mm-hmm. And so maybe I was also, carrying with me the fact that, I have the luxury, I can study, right? So this is what I should do. It's such a luxury.
So young people mostly should be curious.
Yeah. The students, I think, I would always tell them, follow your curiosity and follow your gut feeling. So it's something extremely challenging for the new generation of kids now, because I would always also say, focus, don't look so much left and right, don't be too much influenced. And me, I was lucky to live in at a time where, there were 3 channels on TV, and I could witness the, a 4th and 5th channel. I could see the beginning of MTV, which was very exciting with the clips. But this was actually creative, and this was not influencing badly. This was actually all the new aspect of new things, but with regard to creativity and opening. And now for the kids of this generation, it's very complicated, because at a very, very early age, they are confronted to so many types of information coming left and right.
And sources of cheap dopamine.
Exactly. And I think I would always say, I think as a kid, or even as an adult, I mean, you need to have your time where you can dream, where you do nothing, where you can regenerate, when you can, and where you can learn to listen again to yourself and to the gut feeling. I mean, if someone has a problem, often, it helps to, let's say, to take the time to be also with oneself and analyze the situation. In life, this is often what happens. Life is it's always ups and downs, and there are always unexpected things, and it's always good to reflect, etc. And I feel that for the kids now, it's very complicated, because they are constantly harassed by things. And I want to tell them, take the time to listen to yourself. Don't be worried if you have some doubts. If you don't know what to do in the future, if it's confusing, because it was also confusing for me. I said that I was more interested in science because I felt it was more something that I would like to do, but I could have been also a dancer. I could also have been a hairdresser, even though I don't think I would have had talents in this, but I could have done a lot of things. And I see a lot of kids now, they are very anxious. They think that it's bad if they don't know right away this, if they are not like this, if they are not like that. And like, be cool, you have...
So they see a form and they try to fit in.
Yeah. And you can make what I find nevertheless good, which was not so much the case in my generation, where, there were in France, at a certain age, you needed to study within this frame, otherwise it was too late to be a researcher, etc., etc. And if you don't study this, then you will not be able to do so. And now I think for the new generation, nevertheless, the great thing is that they can choose to study this. They can change, they can study that, and they can end up doing this, and 2 years later they can do that. So I think it's a huge opportunity for them to, they don't have to worry, because they have the possibility to do different things in their lives without being judged, and with the possibility to do it.
So they have the luxury to be able to find...
I think so. I think it's a question of wanting, because specifically in the future, there will be a lot of demand. We will be in lack of manpower in Europe. So,
they should be curious, focused, and find something they will be patient.
Exactly.
Okay, thank you very much. It was a great pleasure.
No, very good.
I wish you a lot of crispy new ideas.
Thank you.
You probably heard this a billion times before.
No, it's the first time that I hear this. So, you see.
Okay, thank you.
Okay, thank you.
Tak dúfam, že sa vám táto epizóda páčila. Pre mňa osobne to bol veľký zážitok a tento podcast je pre mňa takou formou spomienky na stretnutie s takouto významnou vedkyňou a veľmi zaujímavým človekom, Emmanuelle Charpentier. No ako Jozef bežne hovorí, nájdete nás na všetkých lepších, aj trochu horších podcastových platformách. Máme aplikáciu Vedátor bez trackovania, kde sú všetky články, podcasty, môžete nám tam posielať otázky do podcastu. A idete niekde dlhšie autobusom alebo tak, nemusíte byť na sociálnych sieťach, môžete si čítať články o vede, prírode a vôbec u nás. Máme knižku Rozhovory o vesmíre s Norbert Wernerom, takže neviem, kedy presne vyjde táto epizóda, ale je možné, že sa blíži naše živé rozhovory o vesmíre, o vianočnej komete, alebo už boli? Ak boli, tak sorry, nemám pred sebou presne harmonogram. Každopádne ďakujeme, že nás počúvate, veľmi si to vážime a majte sa pekne.
Ak viete o klíme len to, že ju rieši nejaká Greta s komentárom blá blá blá, nájdete si náš klíma podcast denníka SME. Hovoríme v ňom o tom, prečo je táto téma dôležitá a zároveň nebudete počuť, že tu všetci zhoríme, ale hľadáme riešenia a s našimi hostiami sa rozprávame o tom, ako zmenu klímy zastaviť a čo vie preto urobiť každý z nás. A ak vás klíma už zaujíma, zapnite si nás tiež. Vysvetľujeme súvislosti aj hľadáme kontexty. Volám sa Katarína Kozinková a budem sa na vás tešiť. Klíma podcast denníka SME nájdete na sme.sk a môžete ho odoberať vo všetkých podcastových aplikáciách.